everyone. I'm Juliana Lemure, science writer at Genetic Engineering and Biotechnology News, or GEN. In this session of the RFS annual meeting called Precision Medicine, Future Medicine, we're going to focus on the challenges and opportunities in implementing precision medicine. To do that, we'll hear from Kenna Mills-Shaw, the Executive Director of the Institute for Personalized Cancer Therapy at MD Anderson Cancer Center. Before joining MD Anderson, Kenna spent some time at Nature Publishing and was director of the Cancer Genome Atlas Program Office at the National Cancer Institute. Thanks for joining us today, Kenna, and I'm going to pass it over to you.
Awesome. Thank you so much. Hopefully you guys can see my screen. I'm going to be talking today about the implementation challenges and opportunities, as we were just told, and also the importance of decision support in the implementation of precision medicine in the context of oncology. I have a couple of disclosures to go over. I am an editorial board member for Molecular Case Studies. I do receive consultancy fees from GuidePoint, GLG, and Chrysalis. And the technology that I'm going to be discussing in part receives licensing fees from Philips Healthcare, which supports the development of our decision support system at our institution. I first actually want to start with a very specific definition of how we use this concept of precision or personalized medicine. We really use it more in the concept of a precision aspect where we have a specific biomarker generally done or identified in the context of molecular profiling. You might think of next generation sequencing, but it can also be immunohistochemistry, IHC, any form of profiling that can give us a biomarker that tries to distinguish one group of patients from another, primarily in terms of their predictive response to a specific agent, often being a targeted agent. So we could potentially also have prognostic biomarkers or diagnostic biomarkers, but in this context of today's talk, I'm really going to be talking about predictive biomarkers that would suggest to a clinician that we should treat or avoid a specific targeted agent for our cancer patients. The reality is, even though that's a pretty hot topic in the context of oncology care, I'd like to make sure that we all remember that, unfortunately, for most of our patients, we really still aren't using biomarker-driven care. This is just a traditional treatment landscape in prostate cancer presented by Dr. O, but I just wanted to provide kind of this context of, unfortunately, for most of our patients, it looks more like this. There's a series of agents, few of which are directed by a specific biomarker, and if we aren't very proactive or we don't make some choices early on in care in terms of treatment, if we don't catch things early, unfortunately, sometimes these strings of therapies, even though they're standard of care, will still result, unfortunately, in a patient's demise. So we really wanted to set upon a study to look at these patients that didn't have a standard treatment option remaining, focusing just on this patient population and determine whether biomarker identification or molecular profiling could really help us change the paradigm for these patients. So instead of just going through one treatment after another, perhaps use a biomarker to stratify them onto a clinical trial or an off-label treatment. And then if we could do that, we could then implement, if we could show that that would change their outcomes, we could potentially change their standard of care to include biomarker testing and then ultimately use that as part of a standard paradigm. So we did this on 2,000 patients that had gone through their standard of care regimens and no longer had something that was an obvious next step for them. We performed molecular profiling, and I'll talk about which platform we did this on in this context in a moment. And we identified of those 2,000 patients, 789 of them, or just under 40%, had a mutation in an actionable gene. That doesn't necessarily mean that that specific mutation was actionable, but at least the gene at a gene level, we might consider that there was a target for these patients. But unfortunately, despite the fact that we identified about 40% of patients that had a mutation in an actionable gene, only 11% of those patients, or 83 total patients, were put on either a genotype select selected or genotype relevant trial. And these are just distinguished by whether in this context, the genotype selected trials are trials that actually say you must have this specific biomarker to go on a trial, or you must have an activating alteration in this gene to go on a trial, versus trials that were more genotype relevant. So they included a drug which we could make a very clean prediction based on the specific biomarker that might be relevant. So again, just about 11%. And so this might concern you that it was such a low match rate, but the reality is, despite the fact that people were sequencing large portions of the genome, including in this context, the Cornell-Weill Medical School example, where they were doing the exact exome process, they were only matching 5% of their patients. In this publication, we were only using a 50 gene hotspot panel, not a very sexy or forward-looking assay at the time, back in 2012 to 2013. We were matching 11%, again, based on a small portion of the genome. Excuse me, and my animation isn't working for some reason. I apologize for that. But then other studies, including that from MSK Impact, also had about this 11% actionability, going after about 400 genes. So whether you were going after hotspots or hundreds of full-length genes, or even 25,000 genes, we're still only at about the 10%, so low double digits matching rate for precision medicine. I'm going to argue in part it's because regardless of how much of the genome or exome we're sequencing, there's still only on the order of 125 to 150 actionable genes in the context of this very specific definition of biomarker to drug. So we don't necessarily need to be sequencing hundreds of genes to find specific targets in order to act. I would say many of these panels now are on the order of hundreds of genes in order to get enough landscape that we can also capture tumor mutation burden, which some investigators and clinicians are using to predict immunotherapy responses. But otherwise, from a true actionability perspective, we really only need about 125 to 150 genes, including copy number, mutations, and fusions, to get to pretty much the entire oncology actionable genome. So we found, again, about 40% or 789 of our 2,000 patients had a mutation in an actionable gene. Here's the overall percentage of all of the patients and where their mutations were scattered across potentially actionable, non-actionable. So these are things that we knew were mutations that were not actionable or benign, possibly, but others that were potentially germline, because we were only sequencing somatic alterations at this time, or cancer mutations at this time. We were only sequencing tumor, not germline. And we had another, about a third actually, that had no mutation at all found. So they were quiet. Now, the low matchability or matching rate of these patients was not because we did not have a clinical trial available. In fact, every circle that you see is a gene that we considered actionable, as well as a gene that had a clinical trial available over the time period. So it could have been a genotype selected or a genotype relevant trial. Regardless, we had a trial available for this patient population during the period of this study. So it wasn't for a lack of opportunities or a lack of pharmaceutical agents available at our institution. So then we started to try to understand why exactly then we were seeing this low, what we perceived as a low match rate of just that 11%. Instead of going through all 789 patients, we focused on a smaller subset. So I'm going to go over a painstaking analysis of about 429 of our patients. These patients had mutations in our highest actionability genes. So these are genes that had level 1 FDA approval types of associations in our database. And so we would have felt like these patients would have had the highest opportunity for our clinicians to recognize the gene, recognize potential drugs or pharmaceutical agents that could have been, again, either off-label or in clinical trials, and would have had the lowest bar in terms of actionability. So of these 429 patients, 75 of them never came back to our institution. This is, I think, an interesting observation. So 1 in 5, approximately, of our patients who had several $100 to $1,000 test never even knew those results were meaningful for them. They never came back to our institution for any kind of appointment whatsoever. So one thought would be, if we could have proactively reached out, told them about these results, could we have regained that patient population and had an opportunity to improve their treatment paradigm at our institution? Of the patients that did come back, we did lose another third approximately to never having another regimen at MD Anderson. So these patients were simply too sick to undergo any kind of care at all. So they were often referred to hospice care or other facilities, but they never had any kind of treatment after this test. So of the 230 patients that had a mutation in an actionable gene, that were able to come back to our institution for at least one new regimen, we looked at those by doing, again, a painstaking review of their medical records. So these weren't paper, but we didn't have a very comprehensive electronic medical record at this time. This is all hand built. So this is literally, manual chart abstraction. And we looked at all 230, and we found that only 70% of the patients that came back that had a new regimen did the clinician actually even discuss the fact that there was a molecular result in the chart. Of those patients, again, about 2/3 had a clinical trial mentioned associated with those molecular results. But interestingly, that means a third didn't. So a third basically didn't have the results mentioned, and a third that even did have the results mentioned, again, the clinician didn't know or wasn't aware of clinical trials available for them. But of the clinicians that had patients that returned, that were well enough to get another treatment of any kind, that knew where the results were found, that knew what clinical trials were available for those patients, 60% of those clinicians were actually able to match their patients to a relevant agent. This is really an important observation because it suggests that we really have the haves and the have-nots in terms of clinicians at MD Anderson, at least. We have the clinicians that have enough knowledge about our clinical trial portfolios and the molecular phenotypes of their patients to match them to those clinical trials. And we have another population of clinicians that might not have that same level of information or knowledge. So it was really apparent to us that if we could come up with an easy to use, very important, easy to use decision support infrastructure that could be coupled in real time to these molecular results in the medical record for our clinicians, that we might be able to improve the matching rate of these patients. So we had to provide a resource that gave them all of our clinicians an understanding of what the genomic alteration meant biologically and clinically, what clinical trials or drugs were available, and those drugs could be also available off-label. We don't have a lot of off-label use at our institution because of the landscape of clinical trials, but that off-label use is obviously another opportunity. And that creation of that resource is really the goal of the precision oncology decision support system at MD Anderson. So we created this system because the reality was that whether our patients come from all over the globe, and they come in hand with a molecular report that sometimes has a type of decision support, but about 90% of our patients come with an internal or in-house NGS report. And we had to make sure that every patient, regardless of how they were getting profiled, had the same opportunity for decision support that was relevant at MD Anderson. More importantly, when we were looking at the types of reports that were coming to us from external vendors, we were realizing that at least by our standards, these types of reports were insufficient to have the quality that we needed to be able to treat our patients and really help our clinicians. So I'm going to just go through a couple of examples of why it really matters to have high quality decision support. So this is a patient that traveled all the way to Houston from the East Coast that came with an FGFR4 FGFR4 mutation that I point out here. And they pointed this patient to 2 specific clinical trials, which we did have available at MD Anderson at the time. And yet our clinician had never treated anyone with an FGFR4 alteration. So they specifically asked PODS, our decision support team, for an assessment of this alteration. And frankly, we never matched FGFR4 at all, any mutation, to davitinib, to this drug that was used in these trials. And so we searched, we redid all of our research, and we went back to the company and we said, okay, we admit we don't have this association. So where are you getting your association from? And the company said, oh, well, we use a crowdsourced online non-vetted database for our decision support. So we determined that crowdsourcing without, references, et cetera, wasn't going to be our level of standard. So these would be examples of opportunities that seemed to be out there online, but didn't have the level of rigor that we would need to implement at MD Anderson. Other vendors, for example, provided this patient with this alteration, calling it a actionable alteration for a specific target, yet this is a known common polymorphism in a germline allele, and this patient did not have germline sequence, so they called it somatic, but it's very likely, especially at 50% allelic fraction, to be a germline allele. So this patient, again, was given likely incorrect information and suggested to go on a clinical trial based on what is a germline polymorphism. And so we, again, of course, suggested that this wasn't a good match for this patient and sent them to a different treatment paradigm for them. Other groups that I say one person's, you heard the saying, one person's trash is another person's treasure. And one other thing that keeps showing up on these genomic reports is a variant of unknown significance, suggesting that we don't know what this means. There's no opportunity based on the biology that we know. And so I say sometimes one person's VUS is another person's actionable alteration. And in this case, we have a patient's report that showed on, I think, page 27, that this EGFR amplification was all the way back on page 27. When we called the company, it was a very high number amplification, which we pursued because there was 2 actually available clinical trials, I believe, with EGFR amplification as an opportunity or biomarker for enrollment. So we were actually able to enroll a patient on a clinical trial that was not mentioned in this report, demonstrating that I think that we need to make sure that information that is actionable isn't lost in appendices of these very highly complicated reports. Moreover, you need a database that's dynamic, not just for the clinical trial availability, which changes, of course, all the time, but also in the context of what we know about these individual variants, right? So back in 2015, this is an example of a variant that was unknown in terms of its functional significance, but in 2017, we re-annotated this as truly activating based on, again, the literature going down to the level of PubMed IDs, of course, of the resources and the research that support this change in categorization. So making sure that your decision support is dynamic and up to date is absolutely critical to being able to provide clinicians with everything they need to appropriately match patients. So you need to do all of this type of decision support around alterations, those mutations, the copy number alterations, the fusions. You need to do it in the context of all of the clinically available drugs, whether those are clinically available on-label, off-label, or in clinical trial. You need to annotate every clinical trial that's available, right, around those genotype biomarker associations. And you need to integrate all of this within your EHR, and to also allow for the N of 1 or individual patient level response type of information as well, in order to provide robust decision support in real time for patients and their clinicians in the oncology space. So we created this decision support system. We provide all of this information in real time in the medical record for our patients and their clinicians. We definitely don't suggest actually that physicians act on variants of unknown significance. So we're pretty conservative. If we don't know, we don't suggest that it's a match. But if there is a possible match with high level amplifications, with mutations, with deletions, we do provide our clinicians with all of that information. At the end of the day, when we redid a study, when we took patients that had a mutation and we provide decision support on top of that, we went from 11% to about 1/4 of our patients getting matched during the period of this study. And that 1/4 of patients that were matched actually ultimately have an improved outcome. So this is what you'd want to see, right? So similar patients in terms of disease status, similar patients in terms of diagnosis, only looking at the patients that were able to come back to our institution and be treated. Again, that patient population that comes back, it's healthy. They're healthy enough to get on some form of drug. And these are the clinicians that were able to match a patient to a trial versus physicians that were unable to match them. They might have gone on a trial, but perhaps not based on their biomarker status. We see that the patients that were matched do outperform their unmatched counterparts in terms of overall survival. And the original publication, so you can see all of the data is here below for you. That said, I'm going to come back. Well, that's obviously good news, what I just suggested. Unfortunately, precision isn't certainty. And so I think we have a lot more work to do in this field to make sure that we don't rely just on single biomarkers. Here's a very old paper that unfortunately still remains very true in terms of an overarching kind of phenotype that we see in clinical trials where we select on a biomarker. While many patients may respond, those responses may not be long-term or not be sustained. And obviously we have some patients, of course, with lower response or even progression, despite being appropriately matched based on their biomarkers. And I think some of that's for obvious reasons, right? We treat our assessments often of actionability on linear biological pathways, despite knowing that cancer is really this interconnected web, much of which we really have not disentangled yet. And so until we can disentangle more, identify more targets, and make more drugs available, not just alone, but in combination, I think we're going to continue to have a great deal of work, not only in precision medicine, but also in terms of decision support as we move forward. I'm going to close out just by thanking the PODS team, the Precision Oncology Decision Support team, past and present, our laboratory that really helps develop some new assays, as well as our data team that helps us bring all of these data together. Thank you.
Oh, thank you so much, Kenna. That was amazing to learn about that study. I have a couple of questions. We have a couple of minutes for questions. So, I mean, what such a sophisticated and comprehensive amount of data that you just showed. How difficult or what are the challenges to thinking about taking some of the work that you just presented and what you've learned through this study and applying it to places that, you know, aren't doing studies like MD Anderson is doing, making a more broad, applicable situation?
Sure. So I think in an ideal world, right, decision support, these databases, these knowledge bases essentially would be completely publicly accessible, and we could all use them regardless of where we were. Clinicians could just, you know, send a PDF of a molecular result and you'd get back all of the local clinical trials that would be available to your patient just based on your zip code and their molecular status. The reality is there's a lot of these databases are partially publicly available, but unfortunately no one has a complete resource out there right now that is completely 100% dynamic and 100% publicly available, in part because it costs money to maintain this. And I think that question of scalability in a dynamic environment is still one of significant challenge. We haven't yet been able to completely transition or even primarily transition to artificial intelligence or machine learning approaches for much of this work. A lot of it still requires human beings being part of the loop. And when you start to think about scaling that across, you know, every alteration, because every day we see a new alteration that has to literally be manually researched, I think it's daunting, not only that, but then also, of course, as we start moving into more and more types of approaches. We're working right now with companies that are doing RNA sequencing. So they're not just sequencing the genes, they're sequencing the transcriptome, the RNA component. So we're seeing whether, one, are the mutations that are seen in the DNA also seen in the RNA? Do we see expression differences? Are we seeing many more fusions in that context? So even how to deal with these, now that transcriptomics is a new concept, but transcriptomics in the clinical perspective is new. So how do we deal with interpreting these types of readouts in the clinical space, and what does that really mean for our patients when we don't always know what X level of overexpression means? Trying to deal with all of that at scale is going to probably, at least for the near term, be left to many more of these research environments. And really what we need to do is improve this level 1 FDA approved matching for our patients that actually have FDA approved drugs. We're still only at about 50% nationwide matching approved agents to the right patients. So I think we got to start there.
Yeah. And to your point about just more genomic data, I mean, the, vision that we'll all end up with our whole genome sequences on our cell phones at some point down the road. I mean, with patients coming in with, you were saying as they're coming in from other clinical sites, but also just coming in with personal genomes. I mean, how do you envision kind of taking what you're working on now and what it's going to be 5 or 10 years down the road from now?
Sure. I think for us, it's still about finding meaning or, signal in the noise. At least in cancer right now, we still only have on the order of, hundreds of targets, hundreds of pieces of data from the genome that we know how to act on. And so I think it's great to have big data, but the reality is there's still only a small amount of data we can act on. And focusing on separating those two concepts, separating what we can act on, what we can do, what we can use from what's still to be discoverable, and being able to dig into the discoverable, the unknown still, and making those data sets shareable, making those data sets available to research so that we can incorporate them into the research paradigm is what's going to be incredible. And what's going to be critical for future endeavors. The reality is on a day-to-day basis, we're not doing whole genome sequencing on every patient that walks in our door, nor are we seeing that yet. Right. But even if we did, very little of that data right now today is going to be actionable. So really separating that, but being able to leverage it for future discovery is going to be very important.
When you just said not everybody gets a whole genome sequence, does everybody get an exome sequence? Is that a standard protocol?
It's not. So even at our institution, we're doing right now, our assay is about, on the order of hundreds of genes. So about 500 genes, you get 500 genes, tumor normal and copy number, and generally you also would get fusions. So, but we're not even doing whole exomes, and we're not doing any of that necessarily on every patient that walks in the door. We really have algorithms that is this patient likely to receive some form of clinical benefit? Is it a prognostic, a diagnostic, or a predictive measure for this patient with this diagnosis at this point in their treatment? So we have patients, right, that start off as, molecular data wouldn't be helpful. Early prostate cancer, we're just going to watch and wait. That's not a patient that necessarily is going to have a molecular test, but that patient could transition over time, depending on their disease progression, to a patient that would be a good candidate. And so we really, frankly, have specific treatment algorithms depending on the patient, depending on their treatment status, depending on their disease status, for what patients are right for molecular sequencing.
Okay, got it. Understood. And genomics in general is kind of known to suffer from a diversity problem. How do you think about, making sure that there's a wide, diverse range of people included in your program?
So from a patient perspective, really our focus has more recently been, so in the past 2 to 3 years, we've been focused on rare tumors. And so this can be rare tumors from the perspective of diagnosis as well as rare patient populations. And so this is more, I wouldn't say that that's true necessarily from a clinical perspective. We're going to sequence you if you're a patient that fits one of these clinical paradigms. We're going to sequence you clinically, and that's part of your clinical care. But from a discovery perspective, a research perspective, trying to fill the gaps that we have from a diversity perspective, that's going to happen more on the research side. And the Rare Tumor Initiative, and now more recently the Mosaic Initiative, are those initiatives that we really try to figure out if we can identify patient populations either by their demographics or by their tumor type that would merit inclusion into these studies so that we can just do a better job in those populations. And that's happening on the research side.
Okay. All right. Terrific. Well, I think that that's all the time that we have today. So that brings us to the end of our discussion and concludes this session. I want to thank you again, Kenna, for joining us and the great talk. So to all of you, stay tuned for our next session in the program called Out of This World with guests from NASA and the Federal Aviation Administration.